you organizers for the opportunity to come and talk about the marching towards 3 R's principles, that is reduce, refine, and replace animal models in research, and how we can improve the pregnancy preclinical trial to translational side of things. I am a reproductive biologist and been working in the field of preterm birth for the last 35 years. And one of the things, I just have to declare some of the conflict of interest I have as required by our university. The frustration that we have here is we do a lot of research in the lab. We find a lot of molecules that are exciting in animal models, as Christine and Dr. Hahn mentioned, that if you look at this, we actually did a systematic review of preterm birth models, animal models. There are over 500 compounds, close to 500 compounds being tested to reduce the risk of preterm birth or delay preterm birth in animal models. If you think about how many of them have translated from that lab to a clinical trial, and you can guess how many are there. In preterm birth, spontaneous preterm birth, close to 500 molecules have been tested as drugs to treat preterm birth in animal models. Successfully tested, published, yes. Dr. Rees is right, zero. Zero work has translated from animal models to clinical trials. So that is frustrating to us. And this is where the Environmental Protection Agency, the FDA, and the NIH, and just recently, the UK regulatory agency as well, came up that we have to start developing humanized models or more relevant human systems. So Christina mentioned about the FDA Modernization Act 2.0. Actually, last year in December, a few months ago, the 3.0 Act was also unanimously approved by the United States Congress and Senate that we should start using more humanized models and reduce the use of animal models. So this is actually becoming a requirement now. So the 3R principles, reduce, refine, and replace animal models. And how can we do that? So it is not just total replacement, but can we minimize them? Can we use a different species that is much more relevant to human studies? Can we refine our experimental techniques, so utilizing new models? The other thing is, can we reduce the overdose that we use in animal models that actually is not really effective? So using reliable and repeatable experiments, can we avoid excessive injuries in animals? Can we use agents that are causing less adverse effects? Can we improve our statistical analysis so that we don't have to use thousands and thousands, but we can use reduced number of animals in this thing? So these are the 3 principles. And Dr. Hahn and Dr. Entek has already mentioned some of the technology that are being developed now that in the reproductive biology, reproductive health field, how we can march towards these 3R principles. So this is just a ChatGPT image, by the way. And if you look at the new approach methods or NAMs to replace animal models, this is the image that you will come up with. And if you think about this, and many of you who are lab-related researchers or even clinician scientists, you are actually using this all. The 3D cultures or stem cell cultures or 2D cultures or biomarkers, human-based studies, these are all actually NAMs. These are new approach methods or animal models. Now, I am using, with the help of Dr. Entek and Dr. Hahn specifically, we are actually developing organoids and 3D printed devices, cell-based assays. We are also using extensive number of organ on chips, as well as in silico or artificial intelligence machine learning tools to better understand the biology, physiology, endocrinology, and how can we translate this into a better clinical trial. So that frustration coming from that bench to bedside is not happening in our field. Can we improve that using these things? So multiple things. This is just I sent the slides last week. So this is 2 weeks ago. Several things happened. If you think about in the NAMS field, the NIH announced about $150 million in funding for NAMS research, And Dr. Antick's group and Dr. Mark Ferrer, who's the PI, is one of the group that has gotten funding from the NIH to part of this funding. This is basically for infrastructure development, not to typically funding opportunities. This is infrastructure development, technology development, and data hub and coordinators. And just like the MPRINT that you have here, data hub and data coordination center development. In addition to that, the Foundation NIH has also is coming up with additional funding opportunity, and you can see preterm birth is one of their top priorities in funding. So we are actually working with the FNIH to get that. Actually, we are the group that is selected for that preterm birth research by FNIH, and Dr. Han and myself are working on that one. So why do we have to replace animal models, and why do we have to march towards the 3R principles, or why do we have to adapt the new approach methods or non-animal models? If you think about these endocrine functions in that actually determines that the pregnancy as well as parturition, you can see this systemic progesterone withdrawal is one of the biggest theories in animals that actually leads to, that maintains pregnancy, that leads to parturition. And if you look at the human pregnancy, that systemic progesterone withdrawal does not happen. Progesterone level doesn't change. But what is the drug? And instead, functional progesterone, the functional progesterone withdrawal happens. So what is the drug that we have been using in the field for the last 30 plus years? Progesterone. We still keep giving progesterone, thinking that some of these women will benefit from getting extra progesterone in their body, and they can reduce the risk of preterm birth. As you all know, the 17-alpha hydroxyprogesterone caprate was approved by FDA. It was undergoing clinical trials. It was approved, and it was withdrawn about a year or two ago from the market, citing that it is not efficient. It is not efficacious in reducing preterm birth. So there are no drugs right now in treating preterm birth. So this is why, because one of the reasons, the endocrinological difference. Now, there are several similarities too. When I worked in my lab, we actually found that the human fetal membrane senescence or aging of the fetal membrane is associated with preterm, parturition, and premature aging of these fetal membrane is actually associated with preterm parturition. You can see that exact same mechanism in animal models too. So there are differences, but there are also similarities. So we can clearly use animal models to understand several of the mechanistic features of pregnancy and parturition and preterm birth. However, they're not identical in all aspects. So we have to understand where the differences are and where the similarities are. And if we clearly understand, we should be able to use these animals or reduce the use of animals for a specific context of use. So FDA denied use. So I'm going to give you another example. And this is actually done by an MPRINT investigator, Dr. Majid Constantine, and several, many of you here are also part of this thing. FDA denied the use of pravastatin for preeclampsia. And they're cited several things. One of them is the barrier function, including the unreliable PK data. There are several other things. This is just an example. So we decided to do a PK study using 4 different approaches. So these are in vitro models, 4 different approaches. Pravastatin, our question is pravastatin transport kinetics across the fetal-maternal interface. So I'm going to give you an example of the data. So let's see, one of your postdoc or graduate student is coming up with this data, amount of drug transfer, 5 different methods. These are the data that you're getting in the lab during your review. Fetal-maternal transfer of pravastatin across the barrier. So you can see different numbers. Some of them are similar, some of them are totally far apart. What are these approaches used to generate this data? One of them is ex vivo. The other one is a new approach method in silico. This is in silico, by the way. And another 2 NAMs approach, in vitro approaches. Another one is an in vivo model. So what are these models? The first one is placental perfusion, very traditional placental perfusion model. The second one is a GastroPlus, which is a software that you can buy using, that's an artificial intelligence-based PK predictor. The other one is fetal membrane on a chip that Dr. Han actually talked extensively about this one. And this one is a multi-organ fetal-maternal interface. Again, Dr. Hahn mentioned about the placental interface, fetal membrane interface. Those two combined is this one. And the last one is the mouse model. Now you can look at this data and see, and if you present this data of pravastatin based on these animal model data, you may have used about hundreds and hundreds of mouse to do the study and give it to FDA, and FDA say, nope, we're not approving this drug. You can see why. Because the transfer of the drug, I mean, these are humanized models. The transfer of drug in mouse is almost twice as much and what you are expecting in a human model, actually. This is based on human clinical trial data. So this is one of the reasons. So we tested this, and this is the model that we used with the health doctor, with Dr. Hahn's group made this multi-organ chip. We did this. This is published. We are actually now working on multiple other drugs, and a couple of them are already published, the aspirin and indomethacin. We're just making sure that these models, these new approach methodologies can be used to predict PBPK of drugs so that we can actually reduce and refine and replace the animal models in the future. Now again, there are very few drugs, and Christina actually mentioned about this, very few drugs that are in preclinical trials make it to clinical trials. The success rate is abysmal. Actually, more than 90% of the drugs that are successful that fail in clinical trials. However, if you look at the NIH funding on, almost 50% are going towards animal research. Again, there are positive things about animal. If you wanted to understand many of the basic mechanistic aspects, we do need animal studies because we do not get entire human physiology replicated in a new approach methodology. If you look at the alternative methods, only 8% funding, preventive measures, 27%, health disparities, close to 10%. So by switching to methods based on human biology, we could bring safer and more effective drugs to market in less than time. Again, just a summary of what happens, 92% of drugs that pass animal tests fail in human clinical trials. At 30 plus years, average time from lab to get to a clinical trial, and $2.6 billion that are invested per drug, average cost to develop one new drug. So there are several reasons why this, because species differences, ethical concerns, reproducibility crisis, as I said, the drug transport, especially in pregnancy, the fetal-maternal barrier is so different in animal models versus humans. If you look at the reasons for FDA's denial for drugs based on animal models, poor translation from animals to humans, again, I'm not going to read all those things, species to species variations, differential placental transport kinetics and mechanisms, false positive toxicity signals. Some drugs are very efficacious in animals and do not translate well into human trials, whereas some drugs do not work in animals, but they actually work in humans. That examples also exist. So we really have to draw the line where which model, which method, which drug is beneficial. Dr. Hahn and Dr. Attic has already mentioned about the different NAMS technology that are available, in vitro models. There are organoids and tissue engineered models. There are organ on chip or microphysiological system or lab on a chip. Dr. Hahn extensively talked about this. And there are in silico models. So all these things are now available, new approach methodologies or new non-animal models that are available for us to use to better understand the biology, better do preclinical trials, and hope that we can translate these preclinical positive data into a faster clinical trial space. So I just wanted to give 2 bullet points here. Just wanted you to realize that these new approach methods are not replacement for biology. It is not a replacement for human body. They are human relevant tools designed to interrogate specific mechanism within a defined regulatory context of use. Each of these organ chip or each of these tools have very specific context of use, and I will get to that in a few minutes. So the use of one of the criticisms that we always hear, oh, you don't have the whole physiology on a chip. You don't have the whole biology on a chip. Absolutely true. But that doesn't mean that we can use a wrong physiology, a wrong animal model to validate any of these things. So from my own personal experience, I'm not an engineer, I'm not an organ chip expert, I'm not a 3D bioprinting expert, but my own personal experience, for almost 20 years, I worked with the human fetal membrane or amniochorionic membrane. The only thing that I used to do in my lab is fetal membrane explant culture. So I was in Nashville for almost 20 years. This is all I know. This is all I can do in my lab. This is all I have done for 20 years. I studied well, and with knowing that there's plenty of limitations. So when I moved to an academic institution, we started developing these cell lines. So we have developed almost 15 different cell types from human pregnancy-related tissues, including maternal and fetal. So my lab now will look like, if you get a placenta, this is how my lab will look like. 6 or 7 people sitting in different hoods, isolating different cells from a single placenta, creating primary cells, and then modelizing them. So we did these explants, and then we created the cell lines. We realized that there are several limitations by using either one of them. So we developed animal models. So we developed multiple animal models. I think we are the first one to develop an ascending infection model with live bacteria to study human preterm birth mechanism. Again, several limitations. So that's when about 8-9 years ago, I met Dr. Han, and we started developing these organ chip technology so that we can co-culture multiple cells in the same environment and see whether we can advance this field. So at that time, about 8 years ago, I had a fantastic graduate student, Lauren Richardson, who Dr. Han and I co-mentored, and she's now my colleague at UTMB. She's already a tenured associate professor. She also spent 2 years in Dr. Han's lab doing her postdoctoral work, and she has developed several models for us. And she first developed the amnion and amnion membrane on a chip, a 2-chamber chip, and the chorial decidual. Then she expanded that to an entire human fetal membrane. And now we are developing the so she is NIH funded now, NICHT funded now, to develop 3-trimester placenta. So Christina already mentioned about how she 3D prints the 3 different trimesters of placenta, 3 different trimesters of placenta. What Lauren does in my lab is she is actually developing the placenta. This is actually a multi-organ chip with the decidual in the middle, fetal membrane on one side, placenta on the other side. So Lauren has already developed 3-trimester placenta on a chip. And then we had another graduate student again, now he's also my colleague at the UTMB. He developed the lower uterine segment, the vagina, cervix, and decidua on the chip. So we have used these chips extensively to study different mechanistic aspects, drug trials, so on and so forth. So when we did all these things, we have all these chips available, we realized we are still studying the human pregnancy in silos. One day fetal membrane, another day placenta, another day cervix, another day vagina, another day myometrium and decidua. We cannot do this to study human pregnancy. So what we did is we combined this with the help of Dr. Han's lab, and what we created a human pregnancy on a chip. So we can actually study the entire human pregnancy system, the fetal-maternal interface, starting from the vagina all the way to the fetal brain in one system. And this is a very complex 15 different cell types, includes amniotic fluid chamber, and I'll get to that in a minute. And this is just recently funded by the Marsha Dimes. So we have a Marsha Dimes Perinatal Research Center of Excellence. And we are also proposing this to the foundation, NIH, as one of the top 4 projects I already mentioned. So what is pregnancy on a chip? Pregnancy on a chip is, again, trying to recreate the entire human pregnancy-related organs on a platform, plastic platform like this, starting from the vagina all the way to the fetus. You have the uterus, cervix, placenta, and fetal membrane. So the barriers are here. And then you have the fetal membrane, the fetal amniotic fluid and the fetus. And this is an interchangeable compartment. You can put fetal brain or fetal heart or fetal lung or whatever you wanted to study. That's an interchangeable compartment. So the first thing we did is we compared this. Is this even real? There are 15 different cell types, amniotic fluid, interacting each other. There are immune cells in here. There are vaginal microbiome in this compartment. Is this just a fantasy land? Or is it real? So we compared this with human and non-human primate-derived tissues from each of these organs, and you can see the correlation. in transcriptomic data and proteomic data, we were seeing very high correlation between transcriptome expression and proteome expression, suggesting that this pregnancy on a chip is likely resembling human pregnancy-related factors, and we have done many other things to validate this. What we are doing now is, this is a highly complex system. It's very difficult to do. You need to have seven or eight people to actually load the cells onto this thing. So we are actually developing a complex data set from this and using these multi-omics data and combining that with the electronic health record from the patients, trying to create an artificial intelligence model to see whether we can actually use this for predicting risk of preterm birth, as well as suggesting the clinicians that an effective way of intervening these patients who are at high risk. So this is done by a very high group of people who are actually, this is not Scott Williams, actually, this is Paul Wang from Cedars-Sinai, Paul Zhang from Cedars-Sinai. So sorry about that mistake. So they are actually developing an artificial intelligence platform using the data coming from the human pregnancy on a chip. Now, one last aspect that I wanted to get into is, what is the use of these organs on chip? Dr. Han actually mentioned about the TRACE Center or Translational Research Center. Can we use this for predicting, using as a drug development tool? So when we developed all these chips and tested and published, the NIH came to us, NCATS came to us and say, why don't you develop these as a drug development tool? Again, starting with a two-chamber chip, what we did is we are testing this chip. So this is a decidua in the middle and chorion trophoblast cells in the outside. So it's a fetal-maternal interface. The maternal side has immune cells, and you can actually see the immune cells trafficking through these microchannels from the mother to the fetus and back. So you can actually live monitor these things from the maternal side to the fetal. So if you're putting the drug on the maternal side, can it cross to the fetal side? Will it cause any changes on the fetal side? You can actually test this. These are all published. So we actually, in the first year, we used this chip. And what we do is there is a pipeline. Dr. Han's lab will develop this chip, and his lab will undergo a series of quality control measures of this chip. And then he will send it to me, to my lab, and my lab will take a few drugs that we wanted to test. And we will use these chips using our cells and see whether these drugs will cross the barriers and will cause some changes. So we have developed a standard operating procedure using these chips, using these cells, using these drugs, and now it is going through a validation process. This is again done at Texas A&M University by Dr. Van Roosen's lab. What he will do is he will take all the data and all the things from us, and he will repeat all that experiments in his lab on his own. So this is the validation phase. So once these three phases are done, Dr. Roosen's lab will put together, if the data are replicated, he will put together a portfolio for FDA to consider this as a drug development tool. What has done in last November, our first chip, this two-chamber chip, is the letter of intent that we submitted to FDA has been accepted as a drug development tool. And this is the context of uses. This is very important here. The context of use for this chip is, can we measure transfer from the maternal decidua to the fetal chorion? That is one context of use. If the drug is crossing the fetus maternal to the fetal side, is it causing any cytotoxicity or pro-inflammatory changes either on the maternal side or fetal side? Now, in the big scheme of things, what is FDA looking at? This is a decision tree for FDA to approve a drug. And where is this chip fits? So this is where the reduce, refine, and replace animal model comes. The chip is actually fitting right here. It is a small component in a big decision tree. So you can actually, just by putting the chip here, if this chip is approved by FDA, it actually de-risking the whole process. We can potentially reduce some animal use in this area. You still have to use me. Just keep in mind, none of these chips will create a delivery phenotype. You cannot deliver a fetus on the chip. For that, you really have to have an animal model. So this is where these chips are used for. So we are actually pumping each one of these chips that we have developed in the next few years. We will be submitting letter of intent and a qualification program to the new iStand that is created by the FDA for approving these chips as a drug development tool, which very well may accelerate these preclinical trial to a clinical trials phase in human pregnancy and human pregnancy. So in summary, these are some of the principles that I wanted to share with you. The question is not animal models versus not NAMs. The question is, which model provides the most translationally relevant insight for clinical problem? If you think that it is the animal models will give you the best answer, by all means use it. But if you can reduce or refine or replace some of the animals for specific answer, just like I showed you, a NAM has a use it. The goal is not model replacement, but model relevance. Which model you use and what is the relevance of that model? For example, I can tell you an example. I use animal models in my lab. I use CD1 mouse models and LPS and E. coli for studying preterm birth. Next day, somebody asked me, can we collaborate on preeclampsia? Yeah, I have CD1 animals, I have LPS, I have validated E. coli. Let's use that for preeclampsia tomorrow. Day after tomorrow, somebody else asked me, can we use your animal models for IUGR, FGR? Of course. Yeah, I have animal models, we can study this. Whether you're using NAM or whether you're using animal models, there should be a context of use. There are specific answers that you can give, specific questions that you can answer using animals or NAMs. That we need to understand what that is. Regulatory use requires a clear defined context of use. Whether it is NAM or whether it's animal models, you have to have a context of use. You cannot just use, just because I have a model doesn't mean that it is universal and we can use it for anything that we want. Just realize that these NAM literature is now, this is my last slide. This is the NAM literature is actually now going up. NAM term actually came out in 2021. So the new approach methodologies actually started right around 2010. The NCATS was established in, I think it's 2011. But now you can see that in reproduction field alone, the literature publications are going up in this field. So people are actually adapting to the requirements by FDA, EPA, and NIH. With that, I want to thank you all for your attention and thank you all for having us here. And without the funding and without the support from a great team in our lab and Dr. Hahn's lab, Dr. Mark Ferrer's lab, Christina's mentor, this will not be possible. Thank you.